Every human being begins as a single cell. But how does one cell become trillions? The answer lies in cell division.
Inside almost every cell is a nucleus, containing DNA, the enormous instruction manual that controls life.
During DNA replication, each DNA molecule is copied, creating two identical DNA molecules.
As division approaches, the copied DNA becomes tightly packaged into structures called chromosomes. Before a cell divides, its DNA must be copied so that new cells receive the genetic information they need. Mitosis produces new cells used for growth, repair, and replacing old or damaged cells.
Now the cell is ready to divide. But there are two major ways this happens: mitosis and meiosis.
Let's follow one cell through mitosis, beginning with chromosomes that have already been copied. During prophase, chromosomes condense and become visible, while structures called spindle fibers begin forming. The spindle fibers will eventually help move each chromosome to the correct location. During metaphase, duplicated chromosomes line up along the center of the cell. Spindle fibers attach to the chromosomes, preparing to separate their identical copies. This ensures that each future daughter cell receives an identical set of chromosomes. During anaphase, sister chromatids separate and are pulled toward opposite sides of the cell. The result is two genetically similar daughter cells, each containing the same chromosome number as the original cell. Finally, cytokinesis divides the cytoplasm, producing two daughter cells. During telophase, chromosomes reach opposite sides, and new nuclear membranes begin forming. But producing sperm and eggs requires a very different type of division: meiosis. Meiosis. Meiosis begins with a cell containing pairs of chromosomes, one chromosome of each pair inherited from each parent. This process allows organisms to grow, replace cells, and repair tissues. These matching chromosomes are called homologous chromosomes. They contain the same types of genes, although their versions can differ.
Just like before meiosis, the DNA is replicated, creating duplicated chromosomes.
During prophase I, homologous chromosomes pair up in a unique event that does not occur in ordinary mitosis. Sections of DNA can be exchanged between homologous chromosomes. This process is called crossing over. During metaphase Y, homologous chromosome pairs line up in the middle of the cell. Crossing over creates new combinations of genetic information, increasing genetic variation. This is a major difference from mitosis. Homologous chromosomes separate first. Their orientation is random, creating another important source of genetic variation. During anaphase I, homologous chromosomes separate, while their sister chromatids remain attached.
By the end of meiosis I, the duplicated chromosomes have been separated into two groups.
But meiosis isn't finished. Both cells now enter a second division. The original cell divides into two cells, each containing half the original number of chromosome sets. During meiosis II, spindle fibers form again and prepare the chromosomes for another separation. Now the sister chromatids separate and move toward opposite sides. In metaphase II, chromosomes line up individually along the center of each cell. Finally, cytokinesis divides the cells, completing meiosis.
This second separation resembles what happens during anaphase of mitosis.
During telophase II, new nuclei form around the separated chromosomes. Mitosis involves one cell division, while meiosis involves two consecutive divisions. One starting cell has now produced 4 cells, each containing half the original chromosome number. So what is the biggest difference between mitosis and meiosis? Mitosis generally creates genetically similar cells, while meiosis creates genetically diverse cells.
Mitosis maintains the chromosome number, while meiosis reduces it by half.
Mitosis normally produces 2 daughter cells, while meiosis produces 4 cells.
Mitosis supports growth and repair. Meiosis produces cells used for sexual reproduction.
Independent assortment also creates different combinations of chromosomes in reproductive cells.
Meiosis increases genetic diversity through processes including crossing over.
When sperm and egg combine, their chromosome sets come together to form a new diploid cell.
Together, these mechanisms help ensure that reproductive cells are genetically different from one another. These reproductive cells contain half the chromosome number, allowing fertilization to restore the full number. Together, mitosis and meiosis are fundamental to the growth, maintenance, and reproduction of complex organisms.
Every day, your body uses cell division to replace cells that become old or damaged.
Meiosis, meanwhile, creates the reproductive cells needed to pass genetic information to the next generation. First, DNA is replicated so genetic information can be distributed during cell division. Through these processes, genetic information is copied, reorganized, and passed from one generation to the next. In mitosis, prophase, metaphase, anaphase, telophase, and cytokinesis produce 2 daughter cells. Mitosis is mainly about maintaining and replacing cells. Meiosis is about creating genetic diversity for reproduction. In meiosis, 2 rounds of division ultimately produce 4 genetically varied cells with half the chromosome number. In meiosis, 2 rounds of division ultimately produce 4 genetically varied cells with half the chromosome number. From a microscopic chromosome to an entire human body, these tiny cellular processes have enormous consequences. From growth and repair to reproduction and genetic diversity, cell division is one of life's most remarkable processes. This is Biopulse, where biology comes to life.